Well, first I want to thank everybody for coming to this. I want to thank everybody for coming back from the break. I want to thank NFSDC and NIJ for inviting me to speak at this. It's really a privilege and an honor to speak in the company of the others that are here for this. You're going to see a little bit of a repetition in this, and this is kind of a common theme throughout the week, but there are certain points that we want to make because we feel these are some of the more important points of how the field is going right now. So I apologize if this seems a little bit repetitive. I'll try to blow through some of those things that aren't. Right before this, I thought I was going to be in the unenviable position of being the last speaker, keeping everyone between this room and the beach, dinner, and alcohol, not in that order. But thankfully, John stepped up to the plate and has taken that one for me. So I do have a lot of slides. I have like an hour and a half of presentation. Fortunately, I'm a very fast speaker, so this is going to be much faster than that. If it gets too fast, just raise your hand, tell me to slow down. I'm also very grateful to see that everybody was stocking up on caffeine before this. I'm also stocking up on caffeine for this. So we'll get into this. This is going to be basically talking about different approaches to statistical analysis, but then also some of the factors that affect that approach or those approaches. And there's some common things, there's some differences. We're going to see how these effects change how the different approaches may be calculated. But there are some commonalities between them. All right. So just some disclaimers, some of my own disclaimers. I'm not here on behalf of SWGDAM. I'm not here on behalf of ATF. I do not represent, my views may or may not represent those. My experience is with CPI. I started back in 91. I did DQ alpha, polymarker, D1S80, and back then we were doing CPI. We've recently switched over to RMP for mixtures, and I'll talk about that in a little bit. But most importantly, I'm a biochemist. I'm not a population statistician or geneticist, so my apologies to those people who are if I make some mistakes in here. So just really quick, we've discussed two thresholds. How many labs in this room use two thresholds at the moment? How many labs have one threshold? How many labs have no thresholds? Well, I say that somewhat seriously. I mean, I used to do PowerPlex 1.1 on FM Bio, and there were no thresholds. I mean, basically, if you could see it, you called it, because there really was no quantitative way to measure the strength of a peak, or in that case, a band. So obviously, the world's changed a bit, and we always thought, well, geez, when we go from bands to peaks, this is going to be much easier because now you've got this quantitative value. Well, not so much. So I'm now going to talk about the analytical threshold. Obviously, we discussed different ways that can be accomplished. Stochastic threshold, again, just summarizing, this is the threshold set for a single source sample that if you have a peak that surpasses this threshold, you can be confident that it is a homozygous peak or there's some type of primary mutation that's kind of dropped out. But either way, you're treating it as a homozygous peak. If you have a peak below your stochastic threshold, then obviously at this point, you may have dropout of the sister allele associated with that. One of the key points I have in red with this is that if you are applying your stochastic threshold to a mixture, you need to keep in mind that you could have double dosing or allele sharing so that, let's say your stochastic threshold is 200, that may or may not be still applicable to a peak that has shared alleles between the two contributors, and I'll discuss this in the future. So we saw a presentation a couple times about how we might actually come up with your stochastic threshold. There's multiple ways of doing it. I'm just going to go through the way we did it. I'm not saying it's the right way or the only way. Obviously, there's lots of ways. We basically looked at 1,600 heterozygous loci. We looked at 6 different sources of DNA. We looked at, I believe it was 8 dilutions of those 6 sources, amplified them in triplicate, and then summarized all the data. We took all those heterozygous loci, put them all together, ordered them from, so you look at the pairs of the heterozygous loci, order them from the highest peaks if you have 2 peaks, the highest peak down to the very lowest peak. So we had peaks greater than 2,000 down to heterozygous pairs where the highest peak was down around 50, 100 RFU. So we had this list of 1,600 different loci. We created overlapping bins within that, looked at the average peak height within those bins, looked at the standard deviation within those bins, and then created a curve of the average minus 3 standard deviations. The whole point of this was looking at this peak, and I'll just go straight to it. You end up with this curve that, if you look at the actual raw data, it looks kind of, looks like a Himalayas. But you can create a trend line out of this, and this is kind of what you would expect out of this. The higher RFUs, you're going to have fairly consistent peak balance. When you get down to the low end, your peak balance starts going away. And knowing at the ATF we're going to be looking at very low-level samples, ugly samples, we wanted to see what happened at the low end. So we knew that we couldn't just set a 60% peak height balance, expected peak height balance across the board, because we knew at the low end things were going to go awry. So the idea is this curve is the average peak height balance minus 3 standard deviations, meaning this should capture 99.7% of the events that we see. So there will be some exceptions. About 1 in 333 events may fall outside this range, but this should capture the majority of what we're going to see. And so to come up with our stochastic threshold, you come down here and you follow the curve, and when you get down to about 200 RFU, so if your tallest peak is 200 RFU, we saw that the minimum expected peak height balance would be about 25%. 25% of 200 is 50 RFU. Our analytical threshold just happened to be 50 RFU. So a peak that was below 200 RFU could be associated with a peak that is below 50 RFU, which is our analytical threshold. So at that point, you would be observing dropout. So that's how we came up with our stochastic threshold. And when explaining this to juries or other analysts, whatever, I always like to say, okay, up here at this range, if you have a big vat of jelly beans and you've got 1,000 jelly beans in this vat, 500 of them are red, 500 are blue. Up at the upper end, you're going to take 500 jelly beans out of there and count how many red ones and blue ones there are. And you do that 100 times. Most of those times, you're going to get a pretty close to 50-50 split on that. As you get down closer to here, let's say you're just going to take 100 out. Again, you're going to get, if you do that 100 times, you're going to get pretty close to a 50-50 split every time, but you're going to start seeing sometimes where you've got 40 of one, 60 of another, maybe 35 of one, 65 of another. But for the most part, you can get this bell curve, and for the majority of the time, you're going to get pretty close to 50-50 split. But as you continue down, and you get down to here where you say, okay, I'm going to take 10 out. Now all of a sudden, you could get 8 red ones, 2 blue ones, 6 blue ones, 4 red ones, and you start getting this much greater difference in your peak balance or in the amount of jelly beans you're picking out from each color. And obviously, this isn't a falling off the cliff type of thing. This is a slow curve. So if you start at the top and you say, okay, at this point we're taking 1,000 or 500 jelly beans out, and you start going down the curve and going down the curve and going down the curve, as you get smaller and smaller, let's say, okay, you're going to start taking 500 jelly beans, now take 498 out, 496 out, 494 out. And as you go down, you're going to go down this curve, and you're going to start seeing this slow kind of gradual effect where you're going to get more and more imbalance between those two samplings. And this, that's the whole point of stochastic effects. It's a sampling issue. You see these issues because the sample you're taking isn't truly representative of what was in that vat to begin with. So again, we use this chart, and we actually use this in our casework. We have this chart, and we know what the formula is for that curve. So if we have 2 peaks that are fairly imbalanced, we'll plug the peak height of the taller peak into this the formula of the curve and see if it fits within the minimum expected peak height ratio. And therefore, we're kind of getting away from this single 60% across the board. When we first did this, we actually broke it up into like 4 different sections. So you'd say over 1,000 RFU, we would expect 70% peak height balance. Between 750 and 1,000, you would expect 60% balance. And down the board, well, then we found out that, again, you're still kind of doing this multi-level cliff aspect where if you had one bin that went from 500 to 750, and your next bin went from 200 to 499, if I had a peak at 501 and the minimum expected peak height ratio was, say, 50% for that group, but then you look at peak that was at 499, and the minimum expected peak height ratio for that is 35, now you've got this sudden drop-off. Even though you're only going 2 or 3 RFU, you've got this sudden drop-off to the next level. So that's when we went to this kind of smooth curve and just went in that way. And again, this is just an approximation. It's biology. It's messy. It's not going to be an exact science. One of the things we thought was interesting is the average peak height balance across all these bins, going all the way down to 200 RFU, 100 RFU, ranged from 90% down to 71%. That's not really a huge difference, and I'd be thrilled if we had 71% balance down at 150 RFU, 200 RFU. The key was the standard deviation, and that ranged from 8 to 26. And obviously, if you're doing 3 times the standard deviation, that difference is going to be magnified. And so we want to look at, okay, what happens to our stochastic threshold if we change our method? And so for a little while, we were looking at doubling our injection time. And we looked at that, and we said, okay, we went back and redid all the stuff, and we basically found that if we double our injection time, we have to double our stochastic threshold, which somewhat makes sense. I mean, if you have a sampling where you're down to just a few copies, and you've got your 10 blue were selected out, but only 1 red jelly bean was selected out. If it doesn't show up in the amplification, if it doesn't get amplified, it doesn't matter what you do post-amplification, it still isn't going to show up. And so increasing your detection methods really isn't going to pick up that lost allele if it never amplified. So it kind of made sense that if you're doubling your injection time, doubling your sensitivity, you're going to have to double your stochastic threshold. That's exactly what we saw. And then this would go also for some of the other post-amplification or enhanced detection methods. So post-amp desalting, increased cycle number, increased product loaded on the capillary, be it increased injection time or increased injection voltage. So why do we do stats? I'm one of those people in here that actually did enzyme typing. I did ABO typing. First DNA case I ever did was a DQ alpha case. When we did ABO typing or enzyme typing, 1, if we actually got a result, the prosecution was thrilled. 2, if we got a result that said 1 in 5 people would be included, they were just ecstatic. Nowadays, if we tell a U.S. attorney that we got 1 in 500,000, they're like, well, I thought you did DNA. Where are the numbers here? I really don't know how they actually prosecuted people and got convictions before DNA. But there's a reason we did those stats, and we mentioned this before. We have to put some type of weight on this evidence. If we have a result that includes a suspect, we need some way of determining or giving the jury or trier of fact the weight they should put on this evidence. And it's kind of interesting because this should be non-prejudicial. But I know back in the 90s there was a state that had an appellate court ruling that said, well, you know what? There's too much controversy about stats with DNA, so we're just going to say from now on you can't put a stat with the DNA. You just say you got DNA, it matched the suspect, we're done. And somehow that was supposed to be a better result than actually putting a weight to it. And I mean, it didn't matter if you're doing DQ alpha or if you're doing RFLP. If you got a DNA match, you just tell the jury we did DNA and it matched him. And so we're obviously trying to get away from that now. So the whole reason we're doing stats is we want to differentiate or put some type of weight to these statements where no matter how you word it, the DNA profile obtained from the swab of the knife is consistent with the known DNA profile of John Doe, or John Doe cannot be excluded as a contributor to the mixed DNA profile obtained from the swabbings of the grips of the firearm. And this is kind of my world. One of the other reasons why we do this is because SWGDAM tells us we have to. This recommendation, the guidelines basically say laboratory must perform statistical analysis in support of any inclusion that is determined to be relevant in the context of the case, irrespective of the number of alleles detected and the quantitative value of the statistical analysis. And this gets back to the fact that if it's 1 in 5, if it's 1 in 1,000, 1 in a billion, you still have to, if you're going to report it as an inclusion, you have to put a stat on it so we know if it's a weak match or if it's a very strong match. And again, we're trying to differentiate between a match where you would expect 1 in a million people to have this profile versus 3 people on the jury may have the exact same profile. And so this has come up many times across the country, but if you report that someone is not excluded, then they are inherently included. So you can't just say, well, they're not excluded, but it's so complicated that I'm not going to put a stat to it because I don't want to go there. That is, if you're saying they are not excluded, you're inherently saying they are included. So you still have to put a stat with that so the jury knows, jury or trier of fact knows how much weight to put on that conclusion. Bottom line is, if you can't put a number on it, you probably shouldn't be reporting it. You should just report it as neither included nor excluded, inconclusive, however you want to word it, but you shouldn't report it as an inclusion or not excluded. And I have an old colleague that used to say this for a lot of things, but basically you can't make chicken salad out of chicken crap. If it's a bad profile, it's a bad profile, and you move on. Sorry for your luck, but that's the way it is. DNA is not the only evidence, hopefully, in a case, and it's not really up to us to go out on a limb and try to make something more than what we have and get the science kicked out or cause other problems. So then it comes down to, what are the possible approaches? How are we going to do this? How are we going to calculate the statistic? There's the two main ones, and this is for mixtures. There's the combined probability of exclusion, which I believe is probably the most popular one in the US, also known as random and not excluded. And then there's the likelihood ratio. And then there's this third one it's mentioned in the SWGDAM guidelines, and it's something that we've moved to as kind of a way of moving away from CPI, called the random match probability, which we all know that is, that's what we use in single source, but it can also be applied to mixtures. And kind of looking at these, I don't know, I go back to food because I like to eat. But the combined probability of exclusion is kind of like the peanut butter sandwich of cooking, for stats. It's quick, it's easy, everyone knows what you're talking about. There's some differences. You can go with smooth or crunchy, whatever. But it's quick and easy. You're not going to put a lot of time into the interpretation and the calculation. The likelihood ratio is kind of it's the meal you're going to prepare that's going to take a couple hours to do, and you're probably going to like it in the end, but it takes a lot more effort. I like using the peanut butter sandwich for the CPI because people who are LR, people especially in Europe, would go into anaphylactic shock if they were exposed to the CPI because they have a peanut allergy. So, sorry, John. But, and it kind of goes the other way too, where people who do CPI or random and not excluded look at the likelihood ratio and like, you know what, I'll pass on that. And that's where I was for the longest time, and I'm slowly heading there. And it's more a matter of knowing the merits of each and knowing what you want to accomplish. The problem comes in if you try to change the inherent properties of the method you're using. If you use CPI, but you don't like all the data you're throwing away, if you try to modify the CPI and change the definition of what a CPI is, then you're going to get into problems. So my recommendation always is, if you don't like the stat you're using, the statistical approach you're using, don't change your statistic. Don't change how that statistical approach is used. Change to a different statistical approach that uses that data, but just know you're going to have to use a little bit more, put a little bit more effort into it. And there's actually a fourth category that's coming up that we start getting into this continuous model. All three of these models are that binary. Someone's either included or excluded. But when you get to the probabilistic and these continuous models, that's kind of a fourth approach in there, which I don't even know how to compare that to cooking. But it's more of, luckily, these approaches are going to be using some type of software package that actually does it for you. So it's almost like fast food. You get that really fancy meal. Well, not fast food, but going to a restaurant. You get that fancy meal, but you don't have to spend the 4 hours preparing it. So starting with CPI, theoretically, there's no assumptions. It's simple to calculate, usually. Simple to explain to a jury or trier of fact or U.S. attorney or your boss. Independent of the reference profiles. So you can calculate the statistic theoretically without ever looking at the reference profile. Now, you probably aren't going to go ahead and calculate all these statistics before you get the reference, because why go to all that effort if they're never going to submit a reference sample? Disadvantages, it doesn't make the best use of data, i.e. you're probably throwing away some data. One of the key things from the SWGDAM recommendations or guidelines for calculating the CPE or RMP, any DNA typing result used for statistical analysis must be derived from the evidentiary items and not the known samples. Again, this is going to be true for all of these things, and it's kind of a common theme. You need to be able to go through and look at the evidence and calculate that CPE based on the evidence. It has nothing to do with the reference samples. You can't go peeking at the reference samples and say, okay, well, I'm going to use these loci, and I'm not going to use these loci. The CPE is based solely on the evidence. So, you can only have one CPE calculated for the evidence profile, and it is solely based on that evidentiary sample. And this all needs to be determined, what loci you're going to use, how you're going to, what approach you're going to use needs to be determined prior to ever looking at the reference samples. So looking at just a very simple example, what question does a CPI answer? So in this case, you've got a locus looking at the entire profile. You make the assumption it's a two-person mixture, but obviously with CPI, you don't have to make that assumption. What percentage of the population could be a possible contributor to the observed profile? And Chris talked about this before, but CPI, you're looking at all the combinations or all the genotypes that could be a contributor to this. So if you have alleles A, B, C, and D, you've got the AA, the AB, the AC, the AD, the BB, BC, BD, CC, CD, DD. So you take all those things, you do some math, and it comes out to basically just sum all the allele frequencies and square it. That's the CPI. The other side of that is who would be excluded, as obviously 1 minus CPI, but anyone who is an AE, an AD, a DE, whatever, if you don't have one of the alleles present in those 4, you would be excluded. So the random match probability, this is kind of that in-betweener. As my cooking goes, this would be like kind of spaghetti and meatballs. It takes a little bit more time than the peanut butter sandwich, but it definitely doesn't take as long as a 4-course meal. Advantages, it makes a little bit better use of the data. It's still fairly simple to calculate. It's still fairly simple to explain. And it's still independent of the reference profile. So you can go through all this interpretation and your calculation if you want to before you ever see a reference sample. Disadvantages, and whether or not this is actual disadvantage, but it's one of them may be considered that you have to make an assumption. You have to make an assumption as to the number of contributors. And I would also throw in here that one disadvantage is you're still throwing away some data, especially when compared to a likelihood ratio. So the random match probability, looking at the same example, you've got alleles A through D. What question is it answering? Assuming a specific number of contributors, what percentage of the population could be a contributor to the profile, assuming or with the assumption that it's a 2-person mixture? And so if you're doing that, again, it's very similar, but the only difference between the random match probability in this case to the CPI is if it's a 2-person mixture, you can't have any homozygotes. So it's very similar. You would have the sum of all 4 allele frequencies squared, but then you have to subtract out all the homozygote genotype frequencies. So it's A plus B plus C plus D quantity squared, but then subtract A squared, B squared, C squared, and D squared. And again, I apologize, I'm not a population geneticist or statistician. I realize this wording of the formulas is completely wrong, but I hope we all get the idea. And so basically, we're summing all the heterozygous genotypes in this case. So then the likelihood ratio, I think we've seen a little bit of this, and I'm pretty sure Dr. Buckleton's going to go into this a lot more than I want to. But the advantage is it makes even better use of the data, and to date, this would probably be considered the most, or makes the best use of the data available. It is dependent on the reference profile and the actual calculation, but again, the determination of which loci you should use and which genotypes are possible all can be done and should be done prior to looking at that reference sample. Disadvantages, again, whether or not this is a disadvantage, it's up to you, but assumptions must be made. It is more complex to calculate, and it's more difficult to explain. So likelihood ratio, what is it answering? It's a comparison of 2 different hypotheses, the prosecutor's hypothesis and the defense hypothesis. So here, assuming a 2-person mixture with an unknown second contributor and a suspect profile of AB, the numerator is going to be, given this evidence, what is the probability of obtaining this result under the assumption that the suspect is a contributor? So the numerator is going to be 2CD, with the suspect being AB, so the numerator is going to be 2CD. The denominator is going to be, given this evidence, what is the probability of obtaining the result from 2 random individuals? And this is going to be 24 ABCD. So the key question then is, where the heck did 24 ABCD come from? And again, if you look at all the different combinations, you've got AB with the CD. Then to calculate that, it's going to be 2 times AB times 2CD, which is 4 ABCD. 6 combinations of that, you end up with 24 ABCD. You simplify the likelihood ratio, and you're down to 1 over 12 AB. Simple enough. But again, you're seeing, you're using a little bit more of the data to do this. And on Thursday, we're going to go through hopefully multiple examples looking at different things. We're going to compare what the actual numbers are if you do it as CPI on a profile, if you do random match probability on a profile, and if you do likelihood ratio on a profile. And you'll see how the numbers change with the different approaches. And so if we're calculating the CPE or RMP, any DNA typing result used for statistical analysis must be derived from the evidentiary items and not the known samples. Hitting this a couple times. When the CPE, and this is from the SWIG-DAM guidelines, when using CPE or CPI with no assumptions as to the number of contributors, so again, this is just CPI, CPE, not RMP, with no assumptions as to the number of contributors to calculate the probability that a randomly selected person would be excluded or included as a contributor to the mixture, loci with alleles below the stochastic threshold may not be used for statistical purposes to support an inclusion. And this is a big change, or kind of a big problem, because if you're applying CPI to nice, big stains where you've got high RFU values, this is never an issue. As we're, as a field moving towards touch DNA or low levels of DNA, now we're getting a lot more mixtures where you have the lower portion of the minor components, or we're having results that include much lower RFU levels. Now we're seeing where almost entire profiles, if you're doing CPE, have to be thrown away. But if you're doing an approach like RMP or likelihood ratio, you can actually use those loci, and we'll see a little bit more of that on Thursday. So if you have these two loci, and you want to look at the CPI or the RMP for these two loci, you look at locus 1, what genotypes are possible contributors to this mixture, and for the CPI and the RMP, it's pretty much the same. So you look at locus 1, you've got the AA, the AB, the AC, the BB, BC, and CC. This can be and should be done prior to looking at the suspect profile, but you go through and you look at the locus and say, okay, these are the genotypes that I think are possible contributors to this. And you would do the same thing with the likelihood ratio as well. And that's what Chris was just talking about, is coming up with this list of genotypes that are still possible. And if you look at locus 2, it's exactly like locus 1, and you'd have the same conclusion. Oops. So if you have a suspect who's an AB, BC, he would be included, and the CPI and the RMP stat would be exactly the same. If, however, you have a suspect that's an AB and a CD, then, like we said before, while he's included in the first locus, the second locus, he would have to be excluded. This isn't anything new, but it leads to where we're heading with this next one. So let's say you're going to do the CPI or the RMP on a locus, and this red line is the stochastic threshold. Let's assume all peaks are above our analytical threshold, but in this case you have two loci that are exactly the same. A and B peaks are above your stochastic threshold. The C peak is below your stochastic threshold. So now you try to ask the same question: what genotypes are possible contributors to this mixture? You've got the AA, AB, AC, BB, BC, CC, the same 6 genotypes as before. But in addition to that, you have all the potential genotypes that include that C, because it is below the stochastic threshold. So now if the person is a CD, a CE, a CF, whatever, that person would still be included as a possible contributor to this mixture. And this goes the same for the second locus as well. So again, if you have a person who's an AB or a BC, they would be included. You wouldn't exclude a person based on this. But now if you're doing the CPI, you can't use this locus or either of these loci for statistical purposes. With the RMP, you either have to make the decision up front that you're going to use these loci, but you use some type of statistical method to compensate for that, for the potential loss of the cis allele with the C allele. And you have to do the same with both loci. You have to treat them exactly the same. This decision has to be made before you do the comparison to the reference sample. So if you have a suspect that's an ABCD, the statistical calculation, again, must be based on the evidence profile, not the reference profile. So, where we kind of start getting into trouble is if you have a person or a laboratory that's going to say, okay, you know what, I'm going to do CPI, I compare my reference sample to this profile, and I see that he's an AB, so I'm going to use that first locus. But I'm not going to use the second locus, but I'm not going to exclude him because we just said there could be potential allele dropout associated with that C allele. So I'm not going to exclude him based on that because it's scientifically valid that he could be a contributor. but I'm just not going to use that locus for stats because I'll go neutral and just say I can't use that. The problem is, if you're using that first locus for stats, you've just said that anybody who is a CD, a CE, a CF, CG, whatever, is still a potential contributor to that locus. So by doing this, the CPI based on just the ABC alleles, you're underrepresenting in that statistical calculation who you've already said could be a potential contributor, because you're not including the people who are a CD, a CE, a CF, et cetera. So that's why with this, the CPI, you really can't use either of these loci. With the RMP, you can use both these loci if you go with the 2P rule to account for potential loss of the allele with the C allele. So, but again, you need to do this prior to looking at your reference samples and treat both loci exactly the same. So that kind of leads into, what is the 2P rule? I think we've all heard about the 2P rule. I actually had to do a little bit of research where it came from, because it comes back from RFLP days. But it's basically a way of compensating for zygosity ambiguity. That's one of my favorite terms. But basically, you have a single peak. You're not sure if it's a homozygote or if it's heterozygote. And so you're trying to calculate the statistic or the proportion of people that would compensate or that would have that C allele and potentially paired with any other allele. But where did it come from? And it's been explained to me a couple ways, and the second way I actually did on the back of a napkin after having a couple of beers. But the first way, it's a much easier way of thinking about it. It's just 2PQ. The Q is kind of unknown, so you just put a 1 in there, so now it's just 2P. The second way is if you think of a hypothetical locus that has alleles A through E. And you say, okay, I have this A allele. I know it's either an AA or a homozygote, or it's A paired with any other allele. And so if you write that out, you've got A squared or 2AB, 2AC, 2AD, or 2AE. So those are all the possibilities that go with that A allele if there's potential dropout. And so you start doing a little bit of math, and you get down to it ends up being 2A minus A squared. Excuse me. And back in the olden days, they used to just drop off the A squared because it was such a small number, it didn't really matter. And so it was the 2P rule. So right now, like when we incorporate this, it's 2P minus P squared. But again, this is just the way of compensating for any allele that may be associated with the allele that's below stochastic threshold. But of course, Swigdam addressed this and said, well, that's all great, but you still can't use it with CPI. So that's where 4.6.3 comes from. When using CPE or CPI, loci with alleles below the stochastic threshold may not be used for statistical purposes. That's basically what that's saying. You can't. There's no way of compensating for that potential loss of genetic information below your stochastic threshold with CPI. So then, all right, you start talking about the stochastic threshold, and you've got these peaks below your threshold. It used to be you'd say, okay, well, I've got this peak below your stochastic threshold. It's potentially associated with dropout. It's free for all. And Dr. Buckleton and a few others have done a lot of publications on this and a lot of studies on this. And again, kind of going away from the binary model, but going into a continuous model, which we know this is what it is. I mean, it's biology. It doesn't just go and say, oh, I hit 200. Now it's free for all. Dropout could have occurred. If you have two peaks, one at 198 RFU and one at 51 RFU, and your stochastic threshold is 200, sorry, there's very, very little chance that there really is dropout associated with that peak at 198 RFU. There's a much greater chance that you have dropout associated with that peak at 51. However, there is still a chance that there is dropout associated with that 198. I mean, that's why we came up with the stochastic threshold of 200. Even in our studies, and when we looked at these 1,600 loci, we had one instance where there was a peak at 208 RFUs that was associated with a peak that dropped out. And we could see it. It was at like 44 RFU, but it was below our analytical threshold. So, that's one out of, a lot of loci that we looked at. So it is possible, and that's why we came up with that stochastic threshold. But during your interpretation phase, you've got to be looking at that, wondering, should this person really be included if he is an AB? And I'm only detecting that A allele, and it's at 198 or 199 RFU. And this is where the whole probability dropout comes into play. It's kind of why we're trying to incorporate that into the calculation to correct for that difference. And so this is, there's paper out there saying, 2P rule may not always be conservative, because obviously, if you've got that peak at 198 and your suspect is an AB, and you're including that person, that potential genotype as one of the contributors, he's going to be a little bit miffed that he's still included, knowing that the chances that that 198 peak is associated with dropout is pretty low. So then you get into stutters, and we've covered this ad nauseum. So I'll just skip over that whole paragraph. So and this is straight from the example. So you've got the two major peaks, you've got two minor peaks. Assume two-person mixture. You've got the A and the B alleles. We need to treat that B allele as potential true allele. We've kind of covered that. One little twist on that would be, let's say you have another peak. We'll call it E, sort of. E. Now all of a sudden, if you're doing CPI, you still need to include all 5 of those alleles because you aren't assuming the number of contributors. If you're doing likelihood ratio or RMP, and you, based on the entire profile, you look at that and say, I know this is a two-person mixture. Now all of a sudden, you don't need to include that B allele, because with those two minor and with those other 2 peaks, you know that the minor contributor is represented, so we can assume that that B allele is stutter and move away from that. So that's just kind of one example where the rules change a little bit based on the approach you use. And so you'll hear these terms a lot, modified versus restricted. Modified just means you're making an assumption as to the number of contributors. Obviously with CPI, you're not doing that. With RMP, you are, and then with likelihood ratio, you are. With restricted, this is where you're looking at the actual peak height or peak area information and making some assumptions or making some conclusions based on that information. And so it comes down to restricted versus unrestricted. Unrestricted is all heterozygote genotypes are considered possible. So, and I think this was covered a little bit before, but if you have the 4 alleles, A, B, C, and D, and you're doing a CPI or an RMP or whatever, you could, if it's unrestricted, you would include all 4 of those in your potential CPI. If you're doing at a restricted RMP, which is kind of the point, you look at that and you say, okay, based on our minimum expected peak height ratio, I know that the A and the C can't go together, and I know the B and the D can't go together. And it really comes down to only 2 genotypes are potential contributors to this, an AB and a CD. So then this is another determination that you have to make prior to looking at your reference sample. But what happens if the minor component isn't being detected, or what do you do if you're not sure if the minor component is being detected? And again, from the SWGDAM guidelines, whenever the statistical analysis at a locus is meant to represent all possible contributors to a mixture, if there's a reasonable possibility that the locus or that locus dropout could have led to the loss of an entire genotype, then a statistical calculation should not be performed for that locus. So with RMP and likelihood ratio, as long as you think you have at least partial representation of the minor component, meaning at least one of the alleles, you can still use that locus. If there's a possibility that the complete genotype of the minor component is dropped out, then no one should be using that locus. And this really comes into play in different instances, but in this case, let's say you've got degraded DNA. So you've got a pristine major component and then a minor component that has some degradation to it. And you've got that ski slope effect. So you look at the first locus and you can see that, okay, the minor component's there. You look at the next locus, as the molecular weight of the loci are getting larger, you start noticing that that minor component's getting smaller in proportion to the major. But then you get out to, let's say, D16, D2, whatever, and you see 2 large peaks, but you don't see any other peaks. Are you going to be confident that the minor component's represented there? And if you happen to look at your suspect and you say, well, you know what? Let's say those are the 2 peaks you see are A and B. If my suspect's an AB, and it's like, well, you know what? He's represented there. I can calculate your stat for that. That's not really proper, unless you're very, very confident that it would be. But I'm not sure in this situation I would be confident that the minor component's peaks are represented there. And so this is a determination you need to make before you actually do that calculation and you do the comparison. And if you look at that and say, you know what? I don't think it's possible. I think it is possible that the minor component is dropped out. Then you're not going to use that locus for stats. And what you're basically saying is anyone could be that contributor to the minor component. If you kind of go the other way and say, you know what, I'm confident that the minor component's represented there, it's either an AA, an AB, or a BB, then you're kind of boxing yourself into that conclusion that your suspect better be that, or you better be willing to exclude him if he's not one of those genotypes. So then you get into data below your analytical threshold. So we talked about your stochastic threshold, but now you've got your analytical threshold, 50, 70, whatever your analytical threshold may be. We can't use that for comparison purposes, for statistical purposes, or anything else. But I would kind of proffer that you shouldn't just totally ignore it. If I've got a profile, a low-level profile, and we've got no more than 4 peaks across the entire profile, but I look just below our analytical threshold, and I see peaks at like 44, 45, 48 RFU, and assuming our analytical threshold is 50 RFU, and they have the right peak morphology, they fall within bins. If you're doing an RMP, you're making the assumption that there's 2 contributors. If I look at that and I see this at multiple loci, I'm not going to feel comfortable that this is a 2-person mixture and feel confident that I would want to calculate the stat based on that. So what we would typically do if we saw this would basically say it's not suitable for comparison purposes. Because yes, that is below our analytical threshold, and I can't be confident that those are true peaks, but they could be true peaks. And if that's the case, then we have more than a 2-person mixture, and it makes the whole thing beyond our capabilities to interpret. So then major contributor. German Stain Commission, and again, we've talked about this ad nauseum, but type 2 is really what's kind of interesting here. Type B has clearly distinguishable major and minor DNA components, consistent peak height ratios of approximately 4:1 major to minor across all heterozygous loci, and no evidence of stochastic effects. Ignoring the part about the stochastic effects, the part that stands out is that you have to, in order to be able to separate the major and the minor component, they say that you have to have a ratio of 4:1. We've seen a lot of times where you've got the major component maybe 2:1, 3:1, whatever. And I believe NIST has some data to back this up as well, but what they're basically saying is what happens at lower ratios is you can actually have something that's a major, 3:1 here, but if you look at another locus, they flip on you. So now what was the major at 3:1 is now maybe a 2:1 the other way around, where the B component was the minor before, now it's the major component. So while you may be able to separate the components at one locus, you're not sure what happens at that next locus if that major component is actually from that same person, or did they kind of flip and now what was the minor component is now the major component. So while you may be able to separate those out individually at each locus, if it's below a 4:1 mixture or 4:1 ratio, what they're saying is you probably shouldn't multiply that across the board and assume that whatever you're separating out as the major component at that locus is from the same person that is the major component at the next locus. So this kind of makes things a little bit more interesting because it looks, in some cases you would think, well, geez, this is very obvious. I have across the board, I have 2:1 mixture. Looks very obvious. Why can't I just combine all these? But based on this and some of the other data that we've seen, that you really shouldn't be combining that major component across the board because it will potentially flip like that. And so then a little bit more fun. What I talked about before when we were looking at stochastic threshold is that is based on a single source profile. So knowing that you only have one contributor, if you have both peaks above your stochastic threshold or a single peak above the stochastic threshold, you don't have to worry about dropout. I think Mike touched on this a little bit, but let's say you have a 2-person mixture, approximately a 3:1 ratio of components, and you've got a locus where you have 2 alleles, both alleles are above stochastic threshold. Is it safe to assume you don't have dropout? Because you've got both peaks are above stochastic, this shouldn't be a problem. Well, that's not necessarily true. And again, if you have, and this might actually be the exact same slide, the stochastic threshold was based on a single source, so you have to look at the potential for allele sharing. If 130 RFUs of the B allele is actually from the AB major, which falls within the minimum expected peak height balance with the A and the B at that level, then you still end up having 130 RFU for the minor contributor. 130 RFU is below the stochastic threshold, And so you really still could have allele dropout associated with the minor component or the second component in this mixture. And so it's one of those things we just want to emphasize that if you do have a mixture and all your alleles are above the stochastic threshold, that doesn't mean you can automatically do CPI, RMP, or whatever else. There's still a chance that you could have allele dropout. So as we've kind of gotten into this era that there's a big stress on eliminating bias, there are different ways that you can document what your interpretation actually is prior to looking at your reference sample. And so this is something we've come up with at the ATF laboratory. It's something we developed. It's kind of reinventing the wheel, but if you think of like I-cubed and some of these other things as a race car wheel, this is more of tricycle wheel. This is just a very basic method of documenting what our interpretation is prior to looking at the reference sample. And one thing we do is if we run a sample and for whatever reason we don't get a lot of reference samples, we have a evidentiary sample. It comes in, we run it. It's a ugly two-person low-level mixture. We don't necessarily fill this thing completely out right away. We typically would wait until the reference sample comes in, and then once it comes in, before we run it, we'll go ahead and fill this out and do our interpretation, and then we'll run our reference sample. But we're not going to go through this entire process for every profile we get if they're never going to submit a reference. So this is just an Excel spreadsheet. And basically, each of those sections breaks down each of the loci. And you import a allele table from GeneMapper ID, and it autofills this area right here. So in this case, we're going back to one locus that has 4 alleles, 13, 14, 15, and 16, different balances between them. So it autofills the 13, 14, 15, and 16, saying that these are the alleles that were present. We look at this, say, okay, is there a possible complete loss of genotype? I should be drinking something else. In this case, we say no, it's a two-person mixture based on the overall electrophoregram. So no, there isn't a potential loss, complete loss of genotype, so we leave that alone. And so it autofills then over here. These are the combinations of 4 alleles. So could it be a 13 mixed with a 15, or sorry, a 13, 14 mixed with a 15, 16? We look at this based on our minimum expected peak height ratios. In this case, 13, 14 with 15, 16. We say yes, it can. Can it be a 13, 15 with a 14, 16? In this case, you've got the 14 up here, 16 down here. Looking at our minimum expected peak height ratio, we would say, no, you can't have a 14, 16. So therefore, if you can't have a 14, 16, you can't have the 13, 15. And then, so you put a little n in that box, and then you have the 13, 16 with the 14, 15. Again, 13, 16 fall within the minimum expected peak height ratio, and in this case, the 14, 15 hypothetically fit within the minimum expected peak height ratio. So that is a possibility. So then this is going to autofill this section of the spreadsheet, which just basically gives you the potential genotypes that are contributors to this mixture. So when we get a reference sample, all we have to do is pull it up and look at this, these two columns. So we're saying potential contributors to this are 13, 14, a 13, 16, a 14, 15, and a 15, 16. And then using RMP, basically you're just summing, excuse me, you're summing all the genotype frequencies for each of those. So you've got the 2PQ for the 13, 14, 2PQ for 13, 16, and then it sums all these down to that. So this isn't complicated. It's not auto-filling and auto-comparing all the peak height ratios, things like that. We still have to do that manually. But this is something that we document, and then we have initialed off with the date that this was done prior to looking at the reference sample. Questions? Told you that would be somewhat fast.
You make all that program yourself. Is it just Excel-based?
Yeah, the Bureau decided it's much cheaper to use me than actually buy something that does the exact same thing.
Do you know of programs you can purchase that you can do that, or are you sharing that program for price?
iCubed probably, I'm not going to speak for that, but I believe there definitely are some other software programs out there that do very similar things, if not better. There's also the USACIL has a program that does something somewhat similar. The problem is some of these things, another reason why I want to do this myself is I want to know what was happening kind of behind the green curtain. I know like the USACIL, it makes some assumptions that if you have a mixture of two people, it assumes that a shared peak has equal contribution. So if you have three peaks, A, B, and C, and you've got this B peak that is shared allele, the C peak for one component is at 270 RFU, it's going to assume that the shared portion of that B peak is actually 272 or 270 also. And that's not always the case because obviously you get low levels, you can have wide variation. So I wanted to keep this simple because right now there's nothing out there that I found that could actually kind of compensate for all that variation you're going to see in the peak height balance.
Do you actually stop at a point? I mean, if you have 8 or 9 alleles at a locus, are you going to throw in the flag and say this is not going to be something that we're going to interpret, or are you trying to interpret all levels of mixtures from exploded bomb parts?
No. Basically, once we get past 6 alleles, so if it's more than 3 individuals, we throw in the towel and say we're not doing much with it. And that's because if the only time we see peaks above 1,000 RFU is a reference sample. So a lot of our stuff is low level. Even 3-person mixtures are usually useless for us because it's at such a low level that we really can't be confident that it is just 3 people. Because at some point, during our interpretation, you have to make that assumption that it is a 3-person mixture. And if we can't be confident that it is a 3-person mixture, then we're not going to make that calculation. And that's one of the things, it's kind of brings up a point is, even if you do CPI, I don't think many labs have used the same CPI mentality during the interpretation step. When you're doing the interpretation, you usually are making an assumption as the number of contributors, and you're trying to determine who is or isn't a potential contributor to that mixture. And that's one of the reasons where you move to RMP is because if you're making that assumption during the interpretation stage, why not go ahead and use it in the statistics? Because it's already there. It's already mentally being used. You're already kind of going through the same stuff. You might as well use it in the statistics as well.
Tom,
Yes, sir.
I have one question. If you have a messy mixture where you would throw out, basically that profile, if it's 7, 8 alleles, would you still evaluate it for possible exclusions if a suspect is brought to you?
No, because if we're to the point where we don't feel confident that we have representation or we don't know how many contributors there are, then you're to the point where you're not sure at a particular locus if everyone's being represented, so we wouldn't exclude someone based on that either. So if we get to the point where we're not drawing any conclusions, we're not drawing any conclusions. All right.
Are you using the stochastic threshold at all in determining the number of contributors? If you've got 3 alleles that are above stochastic threshold, but you've got 1 allele below stochastic threshold, in balance with everything else you have in the profile, are you considering the possibility that since you've got 1 allele below stochastic threshold, that there may be other things from contributors you might not be seeing?
Not necessarily. It's more of we're looking at the entire electropherogram as a whole. If we have a bunch of, let's say we only have 4 peaks maximum across the board and they're all at 51 RFU, we're probably not going to assume that's a 2-person mixture. But if we have, the majority of the stuff's up in the 200-300 and we've got a couple, like a peak down at 100 RFU or 75 RFU, then we're still probably going to feel comfortable that that's only a 2-person mixture. And so, it really comes down to what is our confidence that it is a 2-person mixture, and we're not going to go out on a limb. I'm not going to risk my career on one case. So, someone can always come along and say, well, what if it's a 3-person mixture? I'll let them calculate that. But, we're only going to draw the conclusion, calculate those stats if we're confident that it is a 2-person or a single source profile. And this kind of sticking with the RMP theme, we will also, if we make the assumption that it's a single source profile, even if we have 2 peaks but they're below the stochastic threshold, we'll just make the assumption that it is a single source profile and we'll do 2 PQ on that. Whereas I know some labs, if they have any 1 peak or 2 peaks below the stochastic threshold, they won't use that for stats. But once you're making that assumption, then you can use it for stats. If you're just saying this is a single source profile, then if you have 2 alleles, you know that you have complete representation of the genetic data. If you have a single peak, then you can use the 2P with that and accomplish the same thing.
reporting that a mixture of consistent with 2 people or assuming 2 people? Do you put that in your report when you use that statistical interpretation?
In the conclusion, we would say it's consistent with the mixture of 2 people, but then with the we would say it's consistent with the mixture of at least 2 people. In the statistics portion of that, right below that, our report's kind of divided up, but for each sample, we would have kind of what the result is, what the conclusion is, and then if relevant, we would do the statistic right below that. For that, we would have a paragraph prior to it or preceding that would just say, assuming a mixture of 2 individuals, blah, and go from there. How come no one else got this many questions? All right, John, I think we have time for about 100 of those 200.